Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Welcome to the MIT Faculty Forum Online. I'm Pam Belloc. I'm a health and science writer for the New York Times, and I will be the moderator of today's webcast. Today's broadcast is sponsored in part by the MIT Federal Credit Union, MIT Professional Education, and MIT Sloan Executive Education. As a reminder, we welcome your questions during the chat. Alumni and affiliated supporters and friends joining us via Zoom, you can use the Q&A feature that you can find on your toolbar, and you can also upvote questions if you want to have them lift to prominence, and we can try to get to those first. We're going to get to as many questions as we can once our presenters have finished. Today's topic is Alzheimer's disease and new research from MIT that is helping us better understand the most common risk gene associated with Alzheimer's. And I write a lot about Alzheimer's. I can tell you this is going to be a really fascinating presentation. Our guests are Professor Li-Huei Tsai, Picower Professor of Neuroscience in the Department of Brain and Cognitive Sciences and Director of the Picower Institute for Learning and Memory at MIT, as well as MIT's Aging Brain Initiative. And Joel Blanchard, Glenn Foundation for Medical Research postdoctoral fellow in the aging research in the Tsai Lab. Welcome, Professor Tsai and Dr. Blanchard.
Thank you, Pam. So I'm Li-Huei Tsai, and as Pam introduced to you, I'm a professor of neuroscience and the director of the Picower Institute for Learning and Memory at MIT. And I'm also heading MIT's Aging Brain Initiative. So before I start, I would like to thank the Alumni Association and of course Pam Belloc for organizing this event, and thank all of you for tuning in to learn about our latest Alzheimer's research. So I'm going to share my screen with you, and at the same time, I'm going to turn off my video just to ensure the robustness of my home Wi-Fi. So can everyone see my screen? Great. So my laboratory seek to understand the pathogenic mechanisms underlying neurological disorders that impact learning and memory, with a particular emphasis on Alzheimer's disease. To date, 5.8 million Americans are living with Alzheimer's disease. 1 in 3 older Americans have Alzheimer's or another dementia. It kills more than breast cancer and prostate cancer combined. In 2019, Alzheimer's and other dementias cost the nation $290 billion, and by 2050, this costs will rise as high as 1.1 trillion. In my laboratory, we take a multidisciplinary approach to investigate all of the brain systems involved in Alzheimer's disease, including neural circuits and brain systems, genes, and the factors affecting their expression, known as epigenomics, and the molecular and cellular mechanisms at play, both in mouse models, but also in cell cultures built from human stem cells. An example of the systems arm of our group is the work on the brain rhythm disruptions seen in Alzheimer's. We have found that we can boost specific gamma frequency oscillations using light and sound at the 40 Hertz frequency. Increasing the power and synchrony of these rhythms across large areas of the brain appears to improve disease pathology and memory in mouse models of the disease. We see reduced amyloid and tau protein buildups, less neuron deaths or neurodegeneration, and improvements in learning and memory. Because this is completely non-invasive, we now have clinical studies testing whether patients with early AD can benefit from this therapy. In the big data work that we do in collaboration with computer scientists, such as Manolis Kallis at CSAIL of MIT, we conduct analysis of large volume of biological and clinical data. We examine the RNA, which is what's produced to express genes from each major cell type in the brain. We found that gender has a huge effect on the biology of the disease in individual brain cells, such as by affecting gene expression in oligodendrocytes, which insulate the wiring of the brain. We found that this insulation of the brain, called the myelination, is centrally involved in Alzheimer's disease. This is important because without adequate myelination, nerve cells don't conduct electrical signals efficiently. A third major approach that we take is to develop new human cell or tissue models from patient-induced pluripotent stem cells. We can use these models to look at Alzheimer's genes and or other new biology identified from our big data approaches in this human system to recreate the human disease in a dish. From there, we can look for small molecules that can reduce or reverse these pathologies to identify new therapeutic directions. Some examples are shown here. Using tissue engineering, we are able to induce myelination of axons in a dish by co-culturing oligodendrocytes and nerve cells. Moreover, we can observe how the immune cells microglia affects Alzheimer's pathology by integrating them into the three-dimensional cerebral organoids. Today's presentation is another example of this very powerful approach. Joel Blanchard in my laboratory did a tremendous job of taking these induced stem cells to make a human tissue model of the blood-brain barrier in the dish. He then recreated Alzheimer's disease features and a condition called cerebral amyloid angiopathy, CAA, and determined how the major Alzheimer's risk gene, ApoE4, promotes this pathology. We are very excited by this work and are looking to see how we can take this forward. Finally, we are also now able to leverage this human blood-brain barrier tissue into many other uses, including trying to understand how SARS-CoV-2 enters the brain vasculature and brain in general to cause some of the brain complications of COVID-19. With that, I will let Joel take it from here.
Hey, thank you, Li-Huei, for the terrific introduction, and thank you all for tuning in. Today, I'd like to share with you some of the approaches we're using to understand Alzheimer's disease and develop new therapeutic opportunities around these insights that we're gaining. And I'll also end with, as Li-Huei mentioned, a brief overview of some of the work that we've recently done to understand how COVID-19 is affecting the human brain and vasculature. I'll start with a brief introduction of Alzheimer's disease. And fortunately, most of us will live cognitively healthy lives into our 80s and 90s. But unfortunately, about 1 in 10 of us will experience cognitive impairments starting around the age 65 or older. And it's women are about 2 times higher risk of having cognitive impairments than men. And currently, this is a really large problem. About 5 million people in the United States have the most common form of dementia called Alzheimer's disease. And it's estimated that by the year 2050, the number of people with Alzheimer's disease in the US alone will nearly triple to about 16 million people. And just to give you a little background on Alzheimer's disease, there are two types of Alzheimer's disease that we talk about. One is the early onset Alzheimer's disease that starts around age 40 and 50 years of age. And this disease accounts for about 5% of all Alzheimer's disease cases. And we know the genetic causes of early onset Alzheimer's disease. For the large or most prominent form of Alzheimer's disease, the late onset, which affects most of 95% of Alzheimer's disease cases and affects people greater than 65 years of age, we really don't know the causes of this type of Alzheimer's disease. And right now there are no effective therapeutics for reversing the cognitive impairments associated with this. We've got some insight into what causes late onset Alzheimer's disease, looking at which genetic variants associate with late Alzheimer's disease. And this gene, ApoE1, is one of the strongest risk factors for late onset Alzheimer's disease. And when we delve into this a little bit more, there are 3 different variants of ApoE gene, and you'll receive one copy from your mother and one copy from your father. And you could either receive ApoE2, ApoE3, or ApoE4. And it turns out that the ApoE4 variant has has a strong correlation with Alzheimer's disease. If you receive one copy of this, the probability of developing Alzheimer's disease later in life is about 3 to 5 times higher. And if you have 2 copies of this, you're nearly 10-fold higher risk of developing Alzheimer's disease. And this is really emphasized in the clinic when we look at patients with Alzheimer's disease. 40 to 60% of all patients have at least one copy of ApoE4. But we don't really understand what Alzheimer's disease is, and it's a major challenge for 2 reasons. One is Alzheimer's disease is a long progressive disease where it can take 20 to 30 years for the actual symptoms and cognitive impairments to develop into what we recognize in the clinic as dementia. The other complicating factor is that ApoE4 is not 100% penetrant. By that, what I mean is there are people that can have even 2 copies of ApoE4 but live cognitively healthy lives into their 80s and 90s. And so this really suggests that Alzheimer's disease could be a multifactorial disease where ApoE4 acts in conjunction with other genetic factors to cause Alzheimer's disease. Or conversely, there might be resilience factors that we don't know about that protect you from the disease-causing aspects of ApoE4. And because we don't really understand what is going on early in the development of Alzheimer's disease, a lot of our therapeutic approaches have focused on the later stages, for example, removing amyloid from the human brain. And these have really shown limited efficacy when we've through clinical trials. And so the approaches we're taking in Li-Huei's lab and at MIT are really to start to understand how genetics and lifestyle promote Alzheimer's disease so that we can move the therapeutic intervention much earlier into the development and hopefully develop more potent and efficacious therapeutics. And so to do this, we've taken a number of approaches, and one of the approaches we're using is comparing postmortem brains from people that have passed away in healthy lives that have no ApoE4 to postmortem brains from humans that have ApoE4 and had Alzheimer's disease. And we're starting to see some remarkable differences. For example, one thing that we're seeing in the brain vasculature shown here is that people with ApoE4 actually have higher expression of the ApoE protein along the vasculature, and the ApoE protein here is shown in green. But in the human brain without ApoE4, they have none of this ApoE protein expression here. And we've looked at a number of cases. This bar graph on the right here shows 4 different individuals that we've looked at, and consistently they had higher ApoE protein. But it's hard to understand how these types of observations, that ApoE4 increases your probability of developing Alzheimer's disease, and that ApoE4 increases ApoE protein in blood vessels, how do they relate to the actual development of the disease? And because we don't actually understand that, we can't develop preventative measures or therapeutic opportunities around these observations. And to do that, we really needed to develop another system to understand how these things relate and are if they are contributing to the development of Alzheimer's disease. And so this scenario is similar to a plane crashing, where after the plane's crashed, you've seen the wings broken off the plane, but you really don't know if the plane crashed because the wing broke off or the wing broke off because the plane crashed. And in aviation, what people have developed is what we call a black box, which allows us to replay the events leading up to the plane crash and start to understand what were the causal events that led to the plane crash. And so in biology and Alzheimer's disease research, we've been really focused on developing that black box to allow us to understand the events leading up to the Alzheimer's disease. And to do this, what we've really leveraged is stem cell biology, neuroscience, tissue engineering, and genome engineering to create what we're calling mini brains on a dish that allow us to replay the disease again and again in the laboratory and understand how different genetic components and environmental components contribute to the disease. And this has really opened up our opportunities to develop new therapeutics. And to really illustrate this today, as Li-Huei mentioned, I'd like to tell you about a story that was recently published in Nature Medicine about the blood-brain barrier. So the human brain is a highly vascularized organ. It's estimated if you stretched out all of the capillaries, veins in the human brain, they would stretch at least 400 miles. And this is really required to provide nutrients and oxygen to the neurons so that they can fire and provide cognitive and functional abilities. And it's really emerged over the past few years that the breakdown of the human brain vasculature leads to cognitive impairments, and a lot of times might be the causal event in Alzheimer's disease. But unlike peripheral vasculature, the human brain vasculature forms a complex structure known as the blood-brain barrier that protects from peripheral pathogens, but it allows nutrients and oxygen to go in. And this blood-brain barrier is really formed through the interaction of three cell types: the brain endothelial cells, the pericytes, and these cells called the astrocytes, which extend processes we call end-feed onto the vascular structure. And so we wanted to know if we wanted to recreate this blood-brain barrier in the laboratory so that we could understand how it breaks down and is altered during neurodegeneration in Alzheimer's disease. And to do that, what we leveraged is human-induced pluripotent stem cells. And so these are cells that we can create by taking skin from a patient and reprogramming them back to the earliest embryonic cell using a combination of transcription factors. And from these induced pluripotent stem cells, or iPSCs, what we can do is generate any cell type in the human body. And here what we did is we generated the brain endothelial cells, pericytes, and astrocytes that make up the blood-brain barrier. And on the right here are images showing that we have the appropriate marker and more shapes for each of these cell types. And so from there, since we had the cells, what we next did is encapsulated them in a hydrogel or 3D environment, which allowed them to interact. And what we observed after about 4 weeks is that the endothelial cells that form the capillaries had actually formed in this dish capillary-like structures shown here in green that are similar to capillaries in the human brain. And one of the things we also noticed is that initially when we put the pericytes into this culture, at week 1, they were evenly dispersed throughout the culture. But after about 2 weeks, because the endothelial cells secrete a growth factor that attracts the pericytes to the vascular space, they are starting to line the vessels as they do in the human brain. And we've done a lot of tests like this to confirm that what we're making in the laboratory, the in vitro blood-brain barrier, has a lot of the hallmarks and anatomical and functional features of the blood-brain barrier in the human brain. And so I don't want to bore you with all those studies, but we think we've been able to create the human blood-brain barrier in a dish. And what we did with it next is asked if we can actually learn anything from this model system about Alzheimer's disease. And I'm going to tell you a story about this pathology that's found, cerebral amyloid angiopathy, or CAA, that's found in the majority of Alzheimer's disease patients. And what this is this amyloid building up, shown here in blue, which is a sticky protein that builds up along the brain vasculature. And what this does to the brain vasculature is it causes it to become leaky, such that it hemorrhages, or it causes it to constrict and blocks nutrients and oxygen getting into the brain, so that there are regions that become ischemic or deprived of oxygen, and neurons start to undergo injury and dysfunction because of this. And so this is a bad pathology that's associated with Alzheimer's disease. And the gene ApoE4 that I mentioned is the strongest risk factor for the CAA pathology. So we wanted to know if we could model this pathology in our induced blood-brain barrier model. And so what we did is we took the amyloid producing cells, which are neurons, from healthy people that have low amyloid and exposed them to the blood-brain barrier. And what we observed is that when we took healthy conditioned media from these individuals, there was no amyloid building up on the vasculature. But when we took conditioned media from AD patients and applied it to the same blood-brain barrier, we started to see this green amyloid building up along the vasculature. And this really suggested to us that we could model this pathology in a dish. And so since we can model the pathology, we wanted to next know if we can model the genetic vulnerability for this pathology and use that to understand why ApoE4 mediates the genetic vulnerability. And so to do this, we used CRISPR-Cas9, which you may have heard about in the news. Essentially what CRISPR-Cas9 allows us to do is create genetically identical stem cells that differ in just one thing. And so we can make stem cells that have ApoE3, which is a non-risk for AD, and we can make identical stem cells that have ApoE4, which is a nearly risk. And so this allows us to isolate specific variables and test their relationship to the pathology we're seeing. And so we did this and made genetically identical ApoE3 IBVs and genetically identical ApoE4 IBVs and exposed them to amyloid. And surprisingly, what we observed is that the ApoE3 had low amyloid along the vasculature, and the ApoE4 had high amyloid on the vasculature. And this really replicates what's seen in the clinic, where ApoE4 individuals have much higher levels of CAA pathology. And so since we can model the genetic vulnerability to CAA, we wanted to know if we could learn something about what is causing the CAA pathology using this model system. And the first thing we turn to is trying to understand which cell types that are expressing ApoE4 are actually causing the pathology. And to do this, we did something that is not possible in the human brain. We mix and match the cellular genotypes. So I've told you we can create an all ApoE3 IBB, and we can create an all ApoE4 IBB, but we can create everything in between, either with one cell expressing ApoE4 or two cells expressing ApoE4. And what this really allows us to do is start to pinpoint which cells are responsible for driving the CAA pathology. And what we observed is that some of the combinations led to low amyloid states that are similar to the ApoE3 IBB, and some combinations led to high amyloid states that are similar to the ApoE4 IBB. And to just summarize this complicated graph, what we observed is that it really didn't matter if the astrocytes or endothelial cells expressed ApoE4. However, when the pericytes expressed ApoE4, this consistently led to a high amyloid state. Conversely, when the pericytes expressed ApoE3, this consistently led to a low amyloid state. And really what this says, and is that pericytes are likely responsible for this increased CA or amyloid pathology. And so now that we've reconstructed the blood-brain barrier in the dish and been able to model the CA pathology and identify the cell type that is responsible for this pathology, we wanted to know, what is it actually doing? What is ApoE4 actually doing in this pericyte to drive this pathology? And to do that, we used some complicated informatics approaches where we can look at the genes inside these cells and identify which genes are different. And it turned out, here's what we call a heat map, where the blue represents low level expression and red represents high level expression. And we observed that there was a striking difference between the ApoE3 pericytes and the ApoE4 pericytes. What really stuck out to us is that ApoE in ApoE4 pericytes is significantly higher than ApoE3 pericytes. And this brings us back to the observation that I introduced in the beginning of the talk, is that we observed in postmortem human brains that along the cerebral vasculature that there are higher levels of ApoE expression in ApoE4 individuals. So through this in vitro model system that we developed, we can start to understand and test why this is and what role it plays in the development of Alzheimer's disease-related pathologies. And really what we found out is, to summarize a lot of work, is that high levels of ApoE in the pericytes are actually leading to this CAA pathology and causing amyloid to build up along the vasculature. And so this really suggests that if we can reduce the ApoE expression in the pericytes, we'll be able to reduce the amyloid pathology. And so we went back to our computational approaches, and what we found is this one signaling pathway that's called NFAT is dramatically upregulated in ApoE4 pericytes. And so we hypothesized that this NFAT signaling pathway was acting like a switch to turn on high levels of ApoE expression, which then subsequently led to the amyloid pathology along the cerebral vasculature. And why this is important is because there are FDA-approved drugs that can block this NFAT signaling pathway. And so if we were correct, we would be able to use these drugs to reduce the levels of ApoE expression. And so indeed, when we tested this in the laboratory, this worked. In ApoE3 pericytes, we observed low levels of ApoE expression, but in the control pericytes, we observed high levels. But when we treated these same pericytes with this drug, cyclosporin A, which inhibits NFAT signaling, we observed reduced pericyte expression, reduced ApoE expression. And so this is an important finding, but we need to go one more step and see if these drugs actually lead to reduced vascular amyloid deposition. And so in the IBB, I've shown you before that there are high levels of this green amyloid building up along the vasculature. And remarkably, what we observed is that when we treated the IBB with cyclosporin or another NFAT inhibitor, FK506, there was dramatically reduced amyloid building up along the vasculature in this in vitro model system. And so we really want to know, this is great, it works in a laboratory, in a dish, but can we actually apply these drugs to an organism and reverse CAA pathology? And so to ask that question, we turned to a mouse model of Alzheimer's disease where the human ApoE4 gene has actually been knocked into the mouse and replaced the mouse's ApoE. So this is a humanized mouse model system. And importantly, in this mouse, around 6 months of age, there's amyloid building up along the vasculature, which is these green or yellowish worms shown here. But when we treated these mice with cyclosporin or FK506, the drugs that we identified, we saw dramatically reduced amyloid building up along the vasculature. So this demonstrates that the discoveries we made in the in vitro model can really reverse the CAA pathology in a living organism. And so it shows promise that it might work in a human. So just to summarize this part of the talk from what I've told you, is that we've reconstructed the human blood-brain barrier in the dish. And we've been able to use that to model a genetic vulnerability to understand a vascular pathology associated with Alzheimer's disease. And this led us to discover that the pericyte plays a role in this pathology. And from there, we were able to understand the molecular mechanisms that underlie it, which led us to a signaling pathway that we could inhibit with FDA-approved drugs and remove the amyloid from the cerebrovasculature in the dish, but also in mouse models of Alzheimer's disease. And we're really encouraged by these results because the two drugs I mentioned, cyclosporin A and FK506, have actually been used in the clinic for a number of years as immunosuppressants following organ transplantations. And what's been observed with people that have been on these drugs is that they have reduced incidence of dementia compared to the general population. So for example, at age 65, 11% of the general population develops dementia, but people on these drugs, cyclosporin or FK506, have a 1% incidence of dementia, and the trend continues all the way up to 85 years of age where the study stopped. So this is really encouraging results, and our study might provide some insight into these observations. But I also just want to underscore that cyclosporin and FK506 are fairly toxic drugs that are given to patients post organ transplant. It's not something that you would want to go out and take to reduce your risk of Alzheimer's disease. But I think the research that we discovered really points to new therapeutic opportunities where we can start to develop less toxic drugs that can probably mitigate some of these risk factors. So for the remaining couple of minutes of this talk, I'd like to tell you about some work that we're starting to do since the rise of the COVID-19 pandemic, and reposition these in vitro models that we're developing to understand how COVID-19 is affecting the brain and the human vasculature. And we're also recently we received brains from COVID-19 patients to look actually at the human postmortem brain tissue to start to understand some of the neurological effects of COVID-19. But using the in vitro models, I'll tell you a little bit about the vascular work we're doing. I think one of the primary factors that's emerged from the clinic is that the virus that causes COVID-19, SARS-CoV-2, is able to infect the vasculature and leads to a lot of vascular associated complications. For instance, in young people that are relatively healthy, there's been an increased incidence of stroke associated with COVID-19. And in older people that have severe COVID-19 symptoms, it's been observed in the clinic that there's a lot of impaired cerebral vascular perfusion or blood flow through the brain of people with COVID-19. And even in infants and toddlers, what's been noted is that there's a rise in this rare disease known as Kawasaki syndrome seen in infants. And what's even more peculiar is this thing called COVID toes, where there's peripheral inflammation in fingers and toes, as shown here down in this picture. And this is thought to due to abnormalities in the microvasculature of peripheral tissue. And so it's really clear that there are vascular complications associated with COVID-19, but there's no understanding of who these vascular complications arise in and the molecular mechanisms underlying them. So we don't have any therapeutic approaches to mitigate things, for instance, like stroke in COVID-19 patients. And so we are rapidly repositioning the vascular systems that I just described in the previous slide of the talk to start to understand how and why vascular pathologies arise in COVID-19. And one of by taking we can generate vasculature from people that have no vascular complications, and we can generate in vitro vasculature from people that do have COVID-19 vascular complications, and combine this with our computational analysis approaches and start to understand the pathways that are different between these two populations of people. And one of these, from this analysis, one of the early insights that we've really started to see is that the cell type, the pericyte that I mentioned earlier, really has high levels of the viral receptor that allows the coronavirus to enter the cell. And we think that genetic differences in the pericyte could influence the vulnerability to these vascular complications. And so we're really excited about this work and looking forward to it progressing. And so I just wanted to end here and thank Professor Tsai for all the support and the vision for all these projects, and also all of our funding for supporting this. The IBBB work that I mentioned was largely funded by Bob and Renee Belfer and the Oscar Fisher Fund, and also the JPP Foundation provided emergency support for our COVID-19 studies. So thank you, and happy to take any questions.
Well, thank you both so much. That was just really interesting. And I can tell that our viewers agree, because there are lots of really great questions. So we're going to do our best to have our speakers answer as many as they can. You can pop more in there. When we reach an end, which I think we probably will before we can get to all the questions, those questions will be sent to our speakers, and they'll have an opportunity to maybe answer them by email or later on. So apologies if we don't get to your questions, and also apologies in advance if I mispronounce your name when I read your question. So I'm going to go through the ones that were upvoted first, the most. This is from Mark Newhausen. Kudos to you on your research. My mother is 91 and has dementia symptoms for almost 5 years, the last 2 years in memory care. I am a big believer in cause and effect. Do we know that the brain symptoms like plaque are causes of Alzheimer's or an effect? And that Dr. Blanchard, that kind of gets to your airplane analogy, I think. It's a good question. Whichever of you wants to jump in, please go ahead.
So maybe I can go first, and because this is a question that really everybody works on Alzheimer's disease, you know, really think about every day. So, you know, basically now, according to the new FDA guidelines, only those patients that show amyloid plaque pathology in the brain are considered Alzheimer's disease patients. If not, even if people have dementia, they are not considered to have Alzheimer's disease. So amyloid plaque definitely is a defining feature for the diagnosis of Alzheimer's. Now, whether it's cause or effect, I think that I am with the view that it can be both. I think Alzheimer's disease is unlikely to be, you know, caused by just one etiology or one particular mechanism. I think that Alzheimer's disease can obviously be caused by different mechanisms and different etiology, you know, different genetic factors, different, you know, lifestyle and other complications. So I think that for certain type of Alzheimer's disease, accumulation of amyloid could be causative. But for some other types of Alzheimer's disease, the amyloid could initially be caused by impairment of some other cellular processes, such as impairment in microglial clearance, of the toxic waste in the brain that then in turn leads to the accumulation. And our recent work on brain rhythms and gamma oscillations, I think also points to this notion. We show that when the power of gamma rhythms is reduced, then that would lead to impairment of microglia clearance and vasculature clearance, and that would then lead to increase in amyloid. And then amyloid abundance would in turn produce other, induce other processes that further exacerbates the pathology. So, so in the end, I believe that there are many different types of Alzheimer's disease. And so that probably call for different kinds of treatments to intervene these various kinds of Alzheimer's.
Dr. Blanchard, do you want to respond to that, or should we move on, take another question just to try to
I think Li-Huei covered it pretty well.
Yeah, yeah. Okay, well, William Shi has asked a couple questions. I'm going to kind of collapse them both, but basically he's interested in the difference between vascular dementia and Alzheimer's disease. And I think this is a question that a lot of people wonder about, and also somewhat evolving, I believe. So which one of you would like to take that?
Joe, would you like to go first?
Sure. Yeah, I think they share a lot of the symptoms in that there's cognitive impairments in both vascular dementia and Alzheimer's disease. I think the origins might be slightly different in that the vascular dementia starts with the vascular system, and it's really diagnosed through MRI imaging where they have white matter hyperintensities, which are a pathology that they see with MRI imaging, whereas the Alzheimer's disease, as Li-Huei mentioned, is really has been reclassified recently as an amyloid disease that has high levels of amyloid in the brain and the peripheral blood.
Dr. Tsai, do you want to continue with that, or should I go on?
So I think Joe basically ended by saying that it's diagnosed differently, right? Vascular dementia is diagnosed based on MRI, looking at white matter hyperintensity with or without amyloid deposition. But for Alzheimer's disease, it has to have amyloid deposition, and Alzheimer's disease, obviously may also have vascular pathology and white matter hyperintensity. So I would say that there could be an overlap between the two diseases, but there are also patients that with vascular abnormalities without amyloid and vice versa.
Okay. Back up here. There's some really good questions about the 40 hertz light and sound therapy. So I'll try to get to a few. So from Ron Cole, I heard about this 40 hertz light effect several years ago. Is there any therapy or preventative process in use? It seems like a very simple, non-intrusive thing to do. Should I get a flashing light? I know you're prepared for that question, Dr. Professor Tsai.
Very good question. Yeah, so definitely there are several small-scale clinical trials ongoing to really test, initially safety of the light and sound 40 hertz therapy in human subjects, and then eventually the effectiveness. So, so I think from what I know, there are human studies going on at MIT and also outside MIT, and this approach appears to be extremely safe in people. There is no any deleterious effect that, as far as we can tell or other people can tell, and now in terms of whether this therapy is going to be effective in either treating Alzheimer's disease or preventing or delaying the onset of Alzheimer's, I think that it will need to be studied in large cohorts of patients in a very well-controlled clinical studies. So, so those experiments are ongoing. The COVID-19 situation really doesn't help because, you know, it prevents person-to-person contact, so it makes the clinical studies particularly difficult. But we are hoping to resume the ongoing studies and evaluation of subjects. So please stay tuned.
Okay, a question from John Kenny. I think another question a lot of people wonder about. Speaking of non-pharmacological interventions, to what extent can diet, nutrition, and other lifestyle steps prevent or delay onset of Alzheimer's? Which one of you would like to take that?
Joe, do you want to start first?
Sure. I mean, I think it's relatively unknown which diet and lifestyle events can prevent it, but I think there are definitely things that correlate with reduced incidence of Alzheimer's disease, such as what the Mediterranean diet, where there's low carbohydrates and more fatty acids, I think has a reduced correlation with Alzheimer's disease. And I think exercise is also something that seems to stave off Alzheimer's disease, so eating healthy, exercising a lot. But the other factor that really is important is maintaining what we call cognitive resilience or cognitive activities. So engaging in things that challenge you a little bit mentally as you age is going to be important to stave off some of the Alzheimer's disease. And we don't really understand why any of these things correlate with lower incidence of dementia or Alzheimer's disease, but we just know that it's important to eat healthy, exercise, and maintain cognitive activity as you age. I think those are good advice for any disease. So,
yeah, so I agree with Joe, and I should also add that I think this is a very important question. Even among the population of older subjects who already show the various pathology in the brain associated with Alzheimer's disease, dementia. And, you know, indeed, majority of people would develop cognitive impairment, but there are people, even with the brain full of plaque and tangles, they can still function very normally. So they're definitely resilient population of people. And you know, we and many other groups are actually very interested to find out, you know, what's the nature of this resilience. So, so we have been conducting, you know, as I introduced to you in the beginning, sort of large data acquisition type of analysis to really identify potential signatures of this resilient population that set them apart from the people who are vulnerable. And we hope that getting this kind of information will help us better understand how lifestyle or diet or activity levels would render the susceptibility to dementia.
Thank you. Okay, we have a question. I'm sure we have several questions about ApoE4, but I'll ask this one from Alyssa Grogan. Does Alzheimer's disease develop with different severity in a person with one ApoE4 allele versus two? I guess she's not asking about the risk, she's asking about the severity, which is interesting.
Joe, do you want to?
Sure. Yeah, I'm not sure there's good epidemiological studies on exactly whether one or two copies alters the severity of Alzheimer's disease. But I mean, what we do have good data on is that the probability of getting Alzheimer's disease is increased with the copy number. So one copy I mentioned is about 4 to 5 times increased probability of getting Alzheimer's disease, and you know, 2 copies, you're up to 10 increased probability of getting Alzheimer's disease. Do you know, Li-Huei, if there's any data on the severity?
So, so I think it definitely affects the age of onset as well. So with 2 copies of ApoE4 on average, it can triggers the onset of dementia, you know, by 5 to 10 years. So instead of, you know, with one allele of ApoE4, most, you know, common age of onset is in the 70s, but with 2 copies of ApoE4, the age of onset could be in the 60s. So, but then again, I want to stress that we humans, you know, our genetic background, we are all very different from each other. ApoE is a major risk factor, but we are also beginning to understand the contribution of other genetic risks. So most likely, in the end, the probability and age of onset of Alzheimer's or other dementia is likely to be the sum of all of these genetic factors that one carry. So I think just, you know, with further study of all of these known risk factors and studying their effects on the different cell types and potential interactions of these genetic factors. For instance, ApoE has already been shown to interact with several other Alzheimer's disease risk genes. So I think this kind of study is going to be extremely informative in educating us, you know, in the end, really, you know, person with what kind of genetic profile is more likely to develop dementia by what age and so on. We are not there yet, but, you know, I think we will be there in the future.
Yeah, I just want to add to that. ApoE4 is a risk factor. It doesn't mean that you're going to have Alzheimer's disease. So even with these genotyping tests like 23andMe, you can identify whether you have ApoE4. There's nothing to worry about there. I mean, I think it increases your risk, but it doesn't mean you'll actually develop Alzheimer's disease. I think we're as Li-Huei mentioned, we're learning that there are multiple factors that go into developing Alzheimer's disease, and we're just understanding them and learning how to prevent them. And hopefully there's great stuff coming down the road on how to prevent it.
Okay, thank you. All right, I'm going to take a question from Robert Lustig, who many of you have probably heard of, is an illustrious nutrition scientist. And he is asking, is insulin resistance a cofactor with ApoE4 that leads to amyloid deposition through altered cellular metabolism, or is it a separate mechanism? Which one of you wants to take that?
Joel?
We haven't looked specifically at insulin and the relationship to ApoE4. I know some companies are developing drugs around insulin for Alzheimer's disease, but it's not an area that we've specifically looked at. I think it's definitely an interesting area to follow up on.
Okay, we have about, I think, 6 or 7 minutes left in our allotted time. I don't know if you guys want to continue over a little bit. It's up to you, but I can just continue.
Sure.
Is that okay? I'll try to continue with as many questions as I can, and maybe we can go a few minutes over. Skipping around a little bit. Okay, hang on a second. Okay, from Candace Joyce Gibson. In those young people who recover from COVID, will they have an increased risk for Alzheimer's disease later in life? That's a really interesting question. I don't know anything about that at all.
So maybe I should start. I can start, and Joe can add after me. I just think that COVID, SARS-CoV-2 is such a new disease. I think we're just in the very beginning of trying to understand. Even just people now with the infection and their clinical presentations, and we're still learning. So I think it's gonna be a while for us to really know whether there's long-term consequences of the infection in terms of the more chronic kind of clinical presentations. But in terms of, I think recently in Alzheimer's field, people have linked viral infection with the risk of developing the disease. It's a very different kind of virus, like a herpes virus and some other viruses, even cytomegalovirus and so on, have been identified. According to some reports, the copy of the viral genome is higher in Alzheimer's disease brain compared to control brain. So now there is this hypothesis that certain viral infection may be linked to Alzheimer's disease. And clearly that's a very interesting new development. I don't know, Joe, you have anything to add?
Yeah, I think it's a great question. I think there's really precedence for these types of events like infection, increasing incidence of Alzheimer's disease later in life. For example, people that have traumatic brain injury or have chemotherapy earlier in their life are at higher risk of developing Alzheimer's disease and dementia. And so I think it's really important area to study, because we've had across the population an enormous number of people that have been infected with this influenza, or the coronavirus, sorry. So I think it's a really interesting area to look out for. And I think with our in vitro models, it's one of the directions that we're heading, is to not only understand how it increases susceptibility for Alzheimer's disease later in life, but in who will it increase susceptibility for later in life, so that we can be prepared for people that are at risk due to this pandemic that's going on.
Really interesting to see what happens there. A question from B. Mayarella. Can the molecular strategies you are applying in organoid culture models be applied instead to whole animal models to create meaningful models incorporating the greater complexity of whole animals?
Joe, that's definitely a question for you.
Yeah, for sure they can be. And I think over the past few decades, we've made tremendous inroads in manipulating experimental mouse models. The trouble there is that the timeline it requires to do this. So a mouse takes about 2 years to grow to age, and in the laboratory we can see Alzheimer's disease around 6 months of age. And so if you extrapolate this out, trying to pinpoint exactly what cell type, you could be looking at timelines close to years and decades to really pinpoint which cell types play the responsible role. Whereas in using these in vitro model systems, it took us a couple months to really pinpoint what cell types were, and then we were able to do the precise experiments to follow up on that observation. And so I think what we're doing will never replace the mouse models, but I think through combining this with the mouse models and post-mortem human brain studies, we're in a better position to really understand Alzheimer's disease and treat it in the near future.
And if I may add some more points, which is, this induced pluripotent stem cells, they're basically taken from human subjects. It can be taken from my skin cells, or Joe's skin cells, or anybody. Of course, patients with Alzheimer's disease, or with carrying ApoE4 genotype or other disease risk genes. So, so this is a tremendous opportunity, amazing opportunity that we are able to study patient-specific manifestation in different cell types that would never be done in animal models.
This is sort of a related question, I think, about, I just lost it, about the human brain dish. From Tyler Pugeda, again, I'm sorry if I mispronounced. Are microglia and peripheral macrophages lymphocytes included in the human brain dish? It seems like neuroinflammation is a significant aspect of Alzheimer's disease.
Great question.
Yeah, so that's really the next direction we're going. And so we built the blood-brain barrier in a dish. And so the next step is to make it more complex and more physiological, more like the human brain. And so we're integrating, we've actually had success in integrating neurons, microglia, and the myelinating cells, the oligodendrocytes, into these cultures. And so we're really looking forward to further exploring the roles of genetics in Alzheimer's disease, using these more advanced cultures that we've recently built.
Okay, a question from Carl. This is also a very good question. How much does your research have application to other brain diseases like Parkinson's?
So maybe I can answer first. So I think that in terms of our work, especially on the ApoE genotype, I think that ApoE, actually E4 in particular, can also increase the risk to develop movement disorders such as Parkinson's disease. And there are definitely overlapping pathways between Alzheimer's and Parkinson's, although the brain cell types and brain circuits affected in these two diseases are very different. So I think just looking at some of the risk genetic factors, there are actually large overlap. So I think, you know, it again, I think stresses, you know, the disease is most likely to be caused by not just cellular molecular processes disruption, but also brain circuit and neural activity contributions to trigger the onset of particular disease. So, so in short, I think that our work could very well have implications for other neurodegenerative diseases.
Dr. Blanchard, do you want to?
Yeah, I totally agree. I think our focus is Alzheimer's disease, but the approaches we're developing are really applicable to any neurodegenerative disease and beyond. And so hopefully there are other people to take that research on and make those discoveries.
Okay, we have a bunch of questions about inflammation. I'm going to try to get to at least one. This is from Claude Gersel. Macular degeneration, which seems to be related of microglia, is closely associated with mutations and complement H. Is there any similar role for complement cascade in Alzheimer's?
Okay, so I can just say a few words first. So now the latest work on neuroinflammation has implicated complement cascade in not just neuroinflammation, but actually on affecting the synapses on neurons. So the synapses are these tiny little contact points between the nerve cells, and they're super important for information processing in the brain and basically underlying the function of the brain. So the complements are implicated in eliminating these important structures in the brain, and therefore affect normal brain function. So I forgot, what is the question now about complement?
I think whether there's some sort of similar complement cascade involved in Alzheimer's. Do we know that?
Yeah. So complement, I think one of the complement genes is actually a genome-wide significant risk factor for Alzheimer's disease. So I think the complement cascade definitely is very much involved. Joe, do you have anything to add?
No, yeah, I agree. I think we see evidence in a lot of cell types that the complement cascade plays a role in Alzheimer's disease.
Okay, this is a really interesting question because I think this is something that is really being actively debated right now in the field. What's more important, amyloid or tau, in development of pathology and cognitive deficits? And how does ApoE4 relate to each of these, amyloid and tau?
Okay, I'll start again. You know, there's very, I think, good evidence, especially in studying the human brain, that the tau pathology is closely associated with cognitive impairment, but not so much amyloid. However, there's also plenty of evidence that showing that the development of amyloid pathology can trigger the development of tau pathology, and there's some certain interactions between the two. And if you look at the timeline in the human brain, one usually first observe the accumulation of amyloid before one detect tau pathology. So it is very likely that the amyloid pathology, you know, is earlier pathology, and tau pathology happens later, and therefore tau has a better correlation with cognitive state. And Joe, do you want to say a few words about their interactions with ApoE?
Sure. Yeah, I think ApoE increases the risk for both amyloid and tau pathology, but we don't understand how either one occurs. So I think that there's an opportunity for discovering the mechanisms there, but also therapeutic approaches. And also tau, one thing to note is that tau is associated with another form of dementia, frontal temporal dementia. And I think because of our clinical diagnosis of dementia over the past decades has been relatively imprecise, a lot of times Alzheimer's disease was a broad term used to encompass frontal temporal dementia, but it's really a different disorder that is solely the tau pathology.
Great. We have several questions about cyclosporin. I'm going to kind of combine them. One person was asking whether cyclosporin, you see it as a treatment of symptomatic Alzheimer's or as a preventive. And a couple people have asked whether transplant patients who are receiving cyclosporin, do we know anything about whether they have any lower Alzheimer's risk?
Yeah, we actually do. So I pointed to a study that's shown that people receiving cyclosporin or the other drug, FK506, actually have about a 10-fold reduced incidence of Alzheimer's disease and dementia. And it's really not, this has been an observational study, so it's really not understood what the molecular mechanisms underlying that are. And so I think some of the results that we found might provide some insight into this. I think it illustrates that these drugs that we identified could potentially be valuable in the clinic, but one thing to note is that they are fairly toxic, and in the case of organ replacements, they're prescribed as life essential immunosuppressants, but in the so that in that case, the toxicity, the benefit outweighs the potential toxicity. But I think in our case, right now, you wouldn't want to take these drugs to offset your risk for Alzheimer's disease, because you might end up with renal failure or other problems. But I think through understanding how they act, we're in a position to discover new therapeutic opportunities that don't have these toxicities and might be even more efficacious or more effective.
Okay, so we're getting close to 10 after 5. I think I'll take maybe one more question. This is also very interesting, and I think this is something that people are studying a lot, and Dr. Tsai, you mentioned this. Any speculation as to why the sex difference? Why are we seeing this more in women than men? I know there are a lot of different theories about this. I'm interested.
Yeah, I think it's a great question. We don't know why. That is a simple answer. But there are a lot of speculations. For instance, estrogen and the sex hormones have been speculated to play a very important role in regulating the different processes in the brain. And also, it is well known, really, many human diseases affect men and women differently. Women are more susceptible to Alzheimer's and multiple sclerosis, and a number of other autoimmune diseases. And men, on the other hand, are more susceptible to autism spectrum disorder and Parkinson's disease. So sex really plays an important role. But I think that our approach to start to interrogate the signatures of each different cell types in the brain in different genders and in different diseases, I think will begin to unveil why men and women have these different kind of susceptibilities to different diseases.
Okay, Dr. Blanchard, do you want to add anything to that?
Yeah, I totally agree. I think for a long time it's been an understudied area, and I think it's just really emerging as something that is very important to study, and it might point to new therapies strategies.
Right. Well, unfortunately, we still have 50 questions left, so there's no way we're going to get to all of them. So I'm gonna give our panelists let them sign off. But all of your questions will be forwarded to them, and I noticed that there are several people who are volunteering to participate in studies, and other people who have questions about testing for either risk genes or other signs of Alzheimer's, and I'm sure they can answer all of those questions. On behalf of the Alumni Association, thank you all very much for tuning into this faculty forum online. Huge thanks to Professor Tsai and Dr. Blanchard for just a fascinating and wide-ranging presentation. It's very exciting research. I certainly am going to want to keep following it and learn a lot more about it. It would be wonderful if we can get some breakthrough on Alzheimer's. Just a reminder that this broadcast will be available on the MIT Alumni Association YouTube channel within a week of today. And I believe the chat window is going to be kept open for about 15 more minutes so that you can add more questions if you have them. Thank you all so much for attending, and thank you so much to our wonderful panelists. I'm Pam Bellick, and I'm happy to join you today.
Thank you. Thank you very much.
Thank you.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.